In this session, we'll be looking at the Biology Paper 2 of AQA Combined Trilogy, and this is the higher tier. This is from the year 2019. It is always a good idea to download the past paper from the description below and have a go before going through this walkthrough. Some students investigated the effect of drinking caffeine on reaction time. They used a drink containing 32.25 milligrams of caffeine per 100 centimeters cubed. This is the method used, so they divide the students into 4 groups A, B, C and D. They measure and record the reaction time of each student using the ruler drop test. In the groups, we have Group A, which has 200 centimeters cubed of water. We have Group B, who drank 200 centimeters cubed of caffeine water. Group C drinks 400 centimeters cubed of the caffeine water, and Group D drinks 600 centimeters cubed of the caffeine water. And then we repeat step 2 after 15 minutes. Describe how to do the ruler drop test. So the method would be 2 students need to carry out the test. Student 1 holds a ruler above the hand of student 2. Randomly, student 1 will let go of the ruler and student 2 has to catch the ruler as quickly as possible. The distance at which the ruler is caught is recorded. Table 1 shows the mass of caffeine taken in by each student. So Group A had 0 centimetres cubed of caffeine in the drink. Group B had 200 centimetres cubed of the caffeine drink. Group C had 400 centimetres cubed of the caffeine drink. And Group D had 600 centimetres cubed of the caffeine drink. So it says calculate the value of X. So what we can do is we can use, there's a couple of different ways, but the way that I'm going to use is if Group B is 200 centimetres cubed of the caffeine drink and Group D is 600 centimetres cubed of the caffeine drink, then for D, it's going to be whatever Group B was, which was 64.5 times 3, because Group D is 3 times as much, and that gives us an answer of 193.5 milligrams, so 193.5. Why did Group A drink water instead of caffeine drink? So this is to be able to compare the effect of the caffeine to no caffeine. Table 2 is used to convert the results of the ruler drop test into reaction times. So the question is, can we estimate the reaction time for a student who recorded a distance of 23 centimetres? So 23 centimetres is between these two distances, which means the reaction time has to be between these two times. So if we look at the difference is 0.009 seconds, which means halfway in between. So if we divide that by 2, it's going to be 0.0045. So if we add that on to 0.212 plus 0.0045, we will get 0.2165. So we would say, as an estimate, a reaction time would probably be 0.217. Any answer between 0.215 and 0.218 should be accepted. Students calculated a decrease in reaction time after the drink compared with before the drink. Figure 1 shows the results for each student, and we've got figure 1 here. So what we can say is Group A to Group D is an increase in caffeine, and the general trend in the results seems to be going up this way. So describe the effect of the mass of caffeine taken on the decrease in reaction time. So as we increase the mass of caffeine, there is an increase in the decrease in reaction time. For 3 students, the decrease in reaction time was negative. Give the reason why the value was negative. So the reason would be that the reaction time actually became larger or greater. What is the range of the results for Group C? So we can see that the range is between 0.01 and 0.18. So just 2 variables that should be controlled in the investigation. So the student should use the same hand each time, and it should be done at the same time of the day so they're equally tired. You can have some other suggestions here for things to control. So we've talked about the same hand, so we could have the same gender of the children. We could have children with the same height. We can make sure that they're all on no medication. We could say that the starting point of the ruler should be in the same place. So there's a number of different things that you could control for this particular experiment. Then we need to explain why the ruler drop test did not involve a reflex action. So it's not a reflex action because it's a voluntary action which involves messages to the brain. There has been a rapid increase in the percentage of carbon dioxide in the atmosphere since the 1960s. Carbon dioxide is a greenhouse gas that contributes to global warming. Name another greenhouse gas. So that would be methane, or we could also have water vapour. Global warming causes climate change, give two effects of climate change. So anything from things like ice caps melting, the sea levels rising, extreme weather conditions, loss of biodiversity, loss of habitats. So any two of those would be correct. Describe how carbon from the atmosphere is cycled through living organisms. Carbon dioxide is removed from the atmosphere during photosynthesis by plants. The carbon makes the glucose, which is then turned into carbohydrates, proteins, and fats. The glucose can also be stored in the plants as starch. When the animals eat the plants, the carbon is converted into proteins and fats in the animals. The animals and plants can both respire to return carbon dioxide into the atmosphere. When animals and plants die, microorganisms feed on the remains as they decay. The carbon is converted into compounds in the microorganisms and they also respire, which means carbon dioxide goes into the atmosphere. Vectors are used in the process of genetic engineering. Which two statements are correct? So the two statements which are correct are vectors are used to insert genes into cells, and vectors are usually plasmids or viruses. Scientists have genetically engineered a variety of wheat to be resistant to herbicide. The herbicide-resistant variety of wheat has a higher yield than the non-herbicide-resistant variety. Explain why. So, if the wheat is herbicide-resistant, when the herbicide is sprayed, only the weeds will be killed. Now, if the weeds are killed, that means that there is more light and nitrates from the soil for the wheat, so they will photosynthesize more and grow more. Give two examples of genetic engineering in use today. Do not refer to herbicide resistance in your answer. So, one of the biggest uses of genetic engineering is for producing medicines, especially things like insulin. We can also say that we can use genetic engineering to produce plants with bigger fruits or which grow faster or which have fruits that have a better taste. Scientists working on the Human Genome Project have now mapped the entire genetic code of humans. Explain one way in which this could be important to people in the future. So, one way could be that we could identify the genes which lead to certain diseases, and if we can identify those genes, we can do work on prevention of those diseases. Animals have adaptations to survive in their environments. These adaptations can be structural, behavioural, or functional. Draw a line from each of the animal adaptations to the type of adaptation it is. So, if we look at the first one, we have male palm cockatoos that use sticks to beat on hollow branches to attract females. So, that's a behavioural adaptation. The harmless hornet moth has black and yellow stripes to look like a bee or a wasp. So, that's a structural adaptation. And the sea spiders have automatic muscle contractions that move oxygen around their bodies. So that's a functional adaptation. Plants also have adaptations. There are more than 28,000 known species of orchid plants. Many orchid plants grow attached to other types of plants, have brightly coloured flowers, produce large quantities of pollen, produce thousands of tiny light seeds. Describe how these adaptations help orchid plants to survive and compete. So the orchid plants will grow on other plants, obtaining water, glucose and minerals from them. The bright colours will attract insects to transfer pollen. By producing large quantities of pollen, there will be more reproduction, and thousands of tiny seeds will travel further because they're smaller and therefore grow in bigger areas. A rare orchid has been found in the mountains of China. The orchid has pale yellow flowers. DNA analysis of the genome shows that it is an ancestral species. All other present-day orchids evolved from this ancestral species millions of years ago. One present-day species has bright purple flowers. Describe how an orchid with bright purple flowers may have evolved from the ancestral species which has pale yellow flowers. There may have been mutations in the ancestral species to produce the purple flower. These were then better adapted to the environment and survived. The purple flower orchids were then able to breed and pass on the allele for the purple flowers. The older ancestral species were not adapted to survive and therefore died out. The DNA code determines the sequence of amino acids which are joined together to form a specific protein. Table 3 shows part of the amino acid sequence for the colour pigmentation protein in 5 orchid species. The rest of the amino acid sequence is the same for all species. So we've got the ancestral, and then we have species A, B, C, and D, and we have the key for the amino acids. Suggest which orchid species is most closely related to the ancestral species and give a reason for your answer. So let's compare the amino acids. So if we look at species A first, we're going to circle the differences. So the second is different. This one is different. And as we go along, we can see this one is different, and that's all for A. If we look at B, we can see that the third one here is different. And then as we go along, we have another difference here, and the other two are the same. If we look at C, we can see that the second one is different. We can see this one is different. We can see that this one is different. And as we go along, we have another difference there, and the other two are the same. If we look at species D, we can see the first one that is actually different is this one here, and the others are the same. So the one that is most similar to the ancestral species is D, because it has the lowest number of differences in amino acids. Some students estimated the population of daisies in a school field. This is the method used. So find a place where some daisies are growing, put the quadrant down, count and record the number of daisies in the quadrat, repeat steps 1 to 3 at 4 different places in the field, calculate the mean number of daisies and use the data to estimate the total number of daisies in the field. And we've got to say which 2 improvements would increase the validity of the method. So the 2 that would increase the validity is repeat for another 10 quadrats. So using a bigger sample size, and also making sure that we're using a random method to place the quadrats. With an improved method, the student calculated the mean number of daisy plants to be 7.65 per quadrat. The student used a quadrat measuring 50 centimetres by 50 centimetres. Figure 3 shows the site and the dimensions of the school field. So we've got 164 metres, 62 and 130 metres there. Now we need to use the map to calculate the population of daisies on the school field, and we've got to give our answers to 2 significant figures. The first thing we're going to do is we're going to divide our field into 2 shapes so we can measure out the area of the field more easily. So we have a rectangle and a triangle. So let's call these 2 areas Area 1 and Area 2. So to work out Area 1, it is going to be 62 metres times the length, which is 164 metres. And to work out Area 2, we're going to have the length, which is 164 metres, times, now the height of that triangle is going to be 130 take away 62, which is 68. So in effect, we're saying 164 times 68 0.8, that would give us a rectangle, but because it's a triangle, we then divide by 2, and that will give us the area of the triangle 2. So the area of the rectangle 1 is 164 times 62, which works out at 10,168 meters squared. The area of the triangle, area 2, is 164 times 68 divided by 2 because it's a triangle, which gives us 5,576. And if we add the two together, we've got 15,744 meters squared. Our quadrat is 0.5 meters times 0.5 meters, which is 0.25 meters squared. And in that 0.25 meters squared, we have 7.65 daisies. So to turn that into meters squared, which is what we've got our area as there, we've got to times 0.25 meters squared by 4. So what that means is to get the number of daisies estimated, we would do our area, which is 15744. We would times it by 4, and then we would times it by the number of daisies in a meter squared. And if we calculate that out, we get an answer of 481,766.4 daisies in the whole of the area, but we've got to give our answer to two significant figures, and with two significant figures, that would be 480,000 daisies. And then the last part of the question, we need to change this into standard form, which would be 4.8 times 10 to the power of 5. And that is your final answer. The student noticed a very uneven distribution of daisy plants in the field. Explain how different biotic and abiotic factors could have caused an uneven distribution of daisies of daisy plants. Use figure 3 on page 20 to help you. So let's have a look at figure 3. Okay, so looking at the diagram here, first of all, we've noticed that we've got areas of trees around. So the trees will reduce sunlight getting to the field, which means there'll be fewer daisies due to less photosynthesis. Also, the trees will take nutrients out of the soil, such as nitrates, which means that there will be less for the daisies to grow, so fewer daisies in that area for that reason as well. We've got a sports pitch here, which means that there's going to be more people walking around the sports pitch, which will kill any of the daisies around that area as well because of trampling. We've got the school building here, which will also reduce the amount of sunlight around that area, which means there could be fewer daisies around that area. We could also have factors like, the school mowing the field, which will reduce the number of daisies in the area that's been mowed. We can have things like near the school building, there's going to be more vehicles, so there's going to be more toxins, which again reduces the growth of the daisies around that area. This question is about homeostasis. Define the term homeostasis. So homeostasis is about controlling the internal conditions of the body to allow the body to function at its optimum. Name the hormone released if the blood glucose concentration falls too low, and that would be glucagon. 2 people were sent to hospital to find out if they have diabetes. This is the method used in the hospital. So you do not drink after midnight, so they're fasting. Then they measure the blood glucose concentration at 9:30, which is here. They drink a glucose solution at 10 o'clock, so that's here. And then they measure the blood glucose concentration for the next 3 hours. So for the next 3 hours, we're going to measure the blood glucose. Person A and person B have diabetes. Describe how figure 4 shows that person B has diabetes. Use the data from figure 4. So we can see person B, we can see even At the fasting levels, it is way higher than a normal blood glucose concentration. Then when they're given the glucose drink, we can see that it shoots up way higher than a normal non-fasting blood glucose concentration. And even after the peak, it is only dropping very, very slowly, and it does not go back to normal levels. Person A and person B had a test to measure the concentration of insulin in their blood when they were fasting, and the table shows the results. Suggest which type of diabetes person A and person B have. So person A will have type 2 diabetes, and this is where their body produces insulin, but their cells do not respond to it. Whereas person A would have type 1 diabetes, and that's where the body is not producing enough insulin to control the amount of glucose in the body. THS has caused the deaths of hundreds of starving children in some tropical countries. The starving children have nothing to eat all day. The starving children then eat many lychee fruits. The lychee fruits contain a molecule which stops an enzyme in the liver from working. This enzyme normally converts fats into glucose. Children who have eaten during the day are not affected by eating many lychee fruits. Starving children may die from eating many lychee fruits, but children who have eaten during the day are not affected, and we need to explain why. The starving children will have very low glycogen stores. They will need their liver enzyme to release glucose from their fats. However, because the lychees stop this enzyme from working, they get no glucose. And if they do not have glucose, they cannot respire sufficiently, so they will die. The children who are not starving will have working liver enzymes and glycogen stores. This means glucagon can release the glucose into the bloodstream, allowing them sufficient respiration.